All right, welcome to this explainer. Today, we are jumping straight into the incredibly fascinating world of molecular dynamics and looking at how it is fundamentally revolutionizing medicine as we know it. We're going to explore a really unique space where biology, computer science, and physics all intersect to solve the kinds of problems that physical experiments, well, they simply can't touch. Okay, so take a look at this headline from HPC Wire. It hits on a massive, everyday problem for patients everywhere. We've all seen those drug commercials on TV, right? The ones that promise amazing, life-saving results, but then they spend half the runtime speed reading a terrifying, mind-numbingly long list of potential side effects. These side effects completely plague prescription drugs. They sometimes force approval agencies to reject them entirely, or they leave patients wondering if the cure is actually worse than the disease itself. Which, naturally leads us to a pretty massive question. How exactly are supercomputers letting us digitally redesign these drugs to just eliminate those unwanted side effects? Basically, how do we take a messy biological problem inside the human body and turn it into a clean digital solution inside a machine? Well, the secret sauce here is molecular dynamics, or MD for short. Think of MD as the ultimate computational microscope. It gives us access to critical information that is literally impossible to get through traditional experiments. Even right now, today, there is no physical experimental technique that lets a scientist continuously watch the motion of every single atom in a biomolecule over time. Molecular dynamic simulations actually bridge that gap. They let us see the invisible. So here is our roadmap for today. We're going to cover the computational microscope, redesigning drugs digitally, solving protein mysteries, how MD works, and finally, the math behind molecular motion. Let's start right in with part one, the computational microscope, seeing the invisible. Wow. Just look at how ridiculously dense and complex these atomic environments actually are. When we simulate a protein, it's not just chilling in an empty, sterile void. To be accurate, these simulations have to account for the target protein in the center, sure, but also the entire surrounding environment. We're talking about the complex lipids making up the cell membrane and the thousands upon thousands of water atoms packed tightly around it. It's a highly crowded, completely chaotic space, which perfectly mimics real biological life. Moving on to section 2, redesigning drugs digitally, fixing side effects. So, how does seeing all this chaos actually help us fix side effects? Well, by using this computational microscope, researchers can simulate exactly where a specific drug molecule binds to its cellular receptor. Imagine looking at an empty molecular pocket on a protein, and then comparing it to a pocket with a drug molecule nestled perfectly inside. By watching that exact binding event happen digitally, scientists can see precisely how the introduction of that drug forces the protein's overall physical structure to change. And this brilliantly illustrates a concept called structural coupling. Get this, binding a drug in one spot actually changes the shape and the binding strength of the protein somewhere else entirely. You can see how introducing different allosteric ligands, like those purple and blue molecules, physically alters the underlying receptor structure. This is an absolute game changer. It gives scientists the exact atom-by-atom data they need to tweak the drug molecule so that it triggers a different, much safer effect. They are essentially engineering out the side effects. Which brings us to section 3, solving protein mysteries, movement and folding. Beyond just redesigning existing drugs, MD simulations are solving massive, dynamic biological mysteries that were previously just impossible to track. For instance, we can literally watch an adrenaline receptor spontaneously shift from an active structure, often modeled in red, to an inactive structure in white. By running these simulations, we can finally describe the actual, mechanical, step-by-step process of how a drug binding on one side of a receptor physically forces the other side of that receptor to change shape and activate. We can even track the entire mind-boggling process of protein folding. Proteins start out in the body as these long, floppy, unfolded strings of molecules. But to actually do their job, they have to fold themselves into highly specific, complex shapes. It's like a giant puzzle. Now, if you just want to predict the final shape, molecular dynamics isn't really your primary tool. But if you want to answer how the protein gets from its unfolded state to its folded state, how it organically manages to find its final shape through an intricate dance of twists and turns, this computational microscope is exactly what you need. All right, section 4, how does it work? The basic algorithm. You are probably wondering how a computer can possibly calculate all of this biological chaos without, just completely crashing. Well, the simulation actually relies on a surprisingly simple three-step loop. Step 1, you divide time into discrete, tiny time steps. Step 2, at each step, you compute the forces acting on every single atom using what's called a molecular mechanics force field. And step 3, you move the atoms just a little bit, updating their position and velocity using good old-fashioned Newton's laws of motion. Then you just loop back to step 1 and do it all over again, millions and millions of times. But the crucial catch here is the scale of those time steps. When we say tiny increments of time, we mean no more than a few femtoseconds per step. A femtosecond, that is 1/quadrillionth of a second, literally 10 to the negative 15 seconds. It's an unimaginably small amount of time. But we have to slice time this thinly because atoms vibrate incredibly fast. If our time steps are too large, the simulation completely breaks down and the atoms will essentially just fly apart.
Finally, Section 5, Math Behind Molecular Motion, Energy and Forces.
To truly grasp how this works, we need to peek at the core physical math. How exactly does the computer calculate step 2 of our algorithm? How does it know what forces are pushing and pulling on the atoms? To do that, the system relies on a potential energy function. The easiest way to wrap your head around this is to picture a 3D hilly landscape. The atoms are basically like little marbles rolling around on this surface. Their physical position determines their potential energy, which we usually mark as U. So let's break down the rules of this landscape. To figure out the force on an atom, you look at how quickly the potential energy changes as you move that atom a tiny, tiny bit in each direction. The faster the energy changes, meaning the steeper the hill, the larger the force. And just like a marble rolling, the force is always going to push the atom downhill toward the lowest possible energy state. When an atom hits the bottom of a valley, a local minimum, the energy stops changing, which means the forces drop to zero. And that brings us to the elegant, foundational equation powering this entire digital universe. Force equals the negative gradient of the potential energy. For a system with thousands of atoms, the computer calculates the derivative of the energy with respect to the x, y, and z coordinates of every single atom. It spits out a massive list of all those forces, plugs them into Newton's equations of motion, along with the mass of the atoms, and calculates their new speeds and directions. That right there is the engine of the computational microscope. So, we've seen how computers simulate biology step by step, calculating forces on a femtosecond scale to literally redesign drugs without those terrible side effects. As our computational microscopes get faster and our mathematical models get even more precise at mapping out these complex, invisible atomic landscapes, it leaves us with an incredibly exciting question. What completely incurable diseases will this technology help us conquer next? Thanks so much for joining me for this explainer, and keep exploring the incredible world of molecular dynamics.